Hello, everyone. Good day, as we say in Denmark. Hope you can hear me well. So we have about almost 100 people registered for this event today. And we have now 13 people in the Zoom. So yeah, let's wait for some minutes before we start. So, by the way, my name is Vang Lê Quý. I'm a senior data scientist at Edinburgh University and working in a special unit called Cloudia, which specializes in the IT service and data science service and data management service for research. So this unit was officially start working from 2018. And this webinar is also part of our activities to promote or facilitate the discussions and exchange of knowledge within Edinburgh University and also between Edinburgh University and external partners or collaborators. So the topic today is actually one of my favorite topics because I have been working with the COVID team in Denmark since March last year.
Hello? Hello, can you hear me?
Yes.
Oh,
Hi,
cool.
Mike. Are you?
Hey, how are you?
Great. Yes, we have my Vella from NVIDIA now also in the Zoom, and he will be the first presenter for today.
Sorry, I'm trying to fix my audio. Van, could you say something else?
Yes.
That's way better.
Yeah, can you hear? Can you hear me?
I can, yeah. Does my microphone sound okay to you?
Yeah, I also need to check my speaker. It's and speaker. Yeah. So I will. I say we wait about for max 2 minutes, and then we get started.
Yeah, that sounds fine.
Yeah. So people can have time to log on.
So I'm trying to figure out how I share my screen here.
Yeah. Yep. Please, shall I stop sharing my screen and then you can.
Yeah, I haven't used Zoom very much, so I'm just trying to understand.
I think in a specific setting, we can have multiple people be able to share the screen. So, but just for now, I will stop sharing and please go ahead.
Okay, view meeting, start share. Okay, this looks promising.
Yeah, at the bottom of the Zoom screen. Yes, I can see the slides.
Okay, terrific. So how is it a big group of people? If it's not a big group of people, we could do a small group.
Last time I checked, it was 92 persons signed up for the event.
Okay. Okay, so that's probably a few too many to do introductions.
Yeah, I mean, it is. It would take time for everyone to introduce, but yes, definitely. I would like to introduce you.
Okay.
Let me see here.
Yeah, you just go ahead and do that, Vang, and then I can start my presentation whenever you're ready.
Yeah. Okay. Now it is 1 p.m. and 2 minutes past 1 p.m. Denmark time, and I could see that there are many people from different time zones. So, good day to everyone. My name is Vang Lê Quý, the senior data scientist at Claudia Aalborg University in Aalborg, Denmark. And today, webinar is the third webinar or events in the series of interesting topics in AI. And I'm really excited to have the two speakers today. The first one is Mike Vella, and he's the solution architect team leader from EMEA area for healthcare in the NVIDIA. And he is also previously engineer at Oxford Nanopore Technology, and he holds PhD in physics. Is that right, Mike?
Yeah, close enough. Computational, computational biology.
From the University of Cambridge, and also undergraduate degree in physics from the University of Bristol. And Mike groups is the group in NVIDIA that helps customers use GPUs to solve problems in healthcare and life science. And the topics today he is going to present and share with us how AI and GPU can be utilized in in genomics and specifically in COVID-19 monitoring. With that, please go ahead, and the screen is yours, Mike.
Okay, great. Can you see my slide?
Yes, and it is somehow it's like dual screen now, so it's not a full screen. Let me see. I can.
Oh, really? Maybe I can do something to fix that. Then hang on. Is that better?
It's the same because I can see your name on the side of the presentation. So it's probably taking, or maybe it's my own screen. I don't know. But how about others? Could you confirm that you can see the full slides? It's not like the shrink down version of the. Yes, someone say yes. So yes, please go ahead then.
Okay, then yeah. If anyone has any issue with the way the slide is showing, then just let me know.
I think it's just a cosmetic thing. I think the screen is big enough.
So I'm going to talk about Oxford Nanopore and sequencing for COVID-19 monitoring. My presentation is going to cover a few topics. It's going to cover, going to touch on artificial intelligence, deep learning, and how it's used in COVID-19 monitoring, specifically in the context of Oxford Nanopore sequencing. And I'm broadly going to talk about Oxford Nanopore sequencing specifically because it is the sequencing technology which uses GPUs essentially. So as you guys probably know, there's 3 major sequencing companies out there: Illumina, Pacific Biosciences, and Oxford Nanopore. And out of those 3, Oxford Nanopore is the one which currently, at least, is using GPUs very extensively. And their technology has found a real role in this recent disease outbreak. So the outline of my talk is I'm going to talk about the genetics of SARS-CoV-2. So I expect a lot of this information is going to be stuff that you guys are already familiar with, but it never hurts to be told something you already know. So hopefully this will be interesting and refreshing to some people. And then I'm going to talk about sequencing for SARS-CoV-2, how DNA sequencing is supporting research, and then finally bioinformatics, including artificial intelligence, and how computers essentially are helping in understanding COVID-19. So let's talk about the COVID-19 pandemic. So most people here are probably sick of hearing this, but COVID-19 is a pandemic which is caused by the SARS-CoV-2 virus. It's a pretty serious respiratory disease, and it's having worldwide spread since around November 2019. All the indications are that it originated in China, but quite quickly spread and seems to have become endemic globally. So what does this viron actually look like? The SARS-CoV-2 is an RNA virus. It's got an outer layer, it's got a lipid bilayer with some embedded proteins, M, E, S, and HE, of which the S protein, the spike glycoprotein, is the most interesting. And the reason is, I say it's the most interesting, it's certainly the most interesting to me and immunologists. And the reason for that, I'll talk about a bit later on, about how its role in how the virus actually replicates. But it's very interesting from an immunology perspective, because since the spike glycoprotein is located on the surface, it makes it a very important target for the immune response, and therefore for developing of vaccines. It's also got a nucleoplasmid with multiple copies of the N protein, which are bound with single-stranded RNA. So, by the way, if anyone has any questions during my presentation, please either just interrupt me or write them in the chat. However, I can't actually see the chat, so Vang, if any questions do pop up, if you could just let me know, because I actually won't notice them.
Yeah, sure. I will monitor the chat.
Thank you. So an RNA virus is, again, this is the material which I suspect everyone here knows very well, but an RNA virus uses RNA to encode genetic information. So unlike most living cells, unlike living cells which encode genetic information as DNA, RNA being used as an intermediary molecule to convert DNA into protein, the RNA is different. The RNA viruses are different. The RNA viruses don't contain any DNA. This is not exclusively the case with all viruses. It's specifically the case with RNA viruses. But their genome is made of RNA, which encodes genetic information, and it produces proteins which hijack the cell's machinery. RNA is not a great material to encode your genetic information, because it's a pretty unstable molecule. But for RNA viruses, they found an ecological niche where this actually works quite well for them. If we look at the genomic organization of the SARS-CoV-2 virus, what's interesting is that quite typical of viruses, really. It's a very small virus. It's 30,000 base pairs long. And this is immediately, when you hear about a virus being an entire organism, if you can think of a virus as an organism, that is, if you think of a genome as being 30,000 base pairs long, for those of you who've worked or are familiar with long-read technology, so that is to say Oxford Nanopore, Pacific Biosciences, this immediately is very interesting because 30,000 base pairs is well within the capability of Oxford Nanopore sequencers to read. And then another interesting thing about this virus, again typical of viruses, is how little DNA is wasted, how little RNA is wasted. Sorry, so there's no junk RNA here. Every single piece of the genome is used to encode genetic information. And if you're a virus, this makes a lot of sense. If you're essentially a small piece of RNA that depends heavily on a sort of parasitic relationship with other organisms to replicate, then there's a huge cost to any kind of unused genomic information. And so that's the reason that the genome of the virus is so information rich. That's why every single piece of it pretty much encodes for a gene. If we look at the SARS-CoV-2 replication cycle, it's typical of coronaviruses, really. So the spike protein binds to cell surface protein in humans called ACE2. And this surface protein is commonly found in the lungs, which I suppose, I don't want to say anything which is scientifically incorrect, but it's plausible to believe that the reason that the SARS-CoV-2 causes such serious respiratory problems is that the spike protein binds to a cell surface protein in humans, which is commonly found on the lungs. I don't know if that's proven beyond doubt, but it seems like a very plausible hypothesis. Once this binding happens, the virus releases RNA, and the RNA is translated into protein by the cell's machinery. And some of these proteins are then used to make more viral RNA. Finally, new virons are assembled inside the Golgi apparatus of the cells and released. And so the cell is affecting, the viron SARS-CoV-2 is affecting human lung cells and using them to replicate. So now I want to talk about sequencing for SARS-CoV-2. SARS-CoV-2 has been sequenced with multiple technologies and labs, and one remarkable thing about this pandemic is how rapidly this virus was sequenced. So the first sequence from Wuhan, and which is believed to be the origin of this virus, was deposited in gene bank on January the 5th. So we don't know, I don't think, exactly when this virus developed, but we do know that very shortly afterwards, it had been sequenced and been made available by Chinese scientists for the world to study. RNA was extracted from the lungs and sequenced with Illumina and BGI sequencers. This was followed by de novo assembly and targeted PCR and Sanger sequencing for filling the gaps. And all the protein sequences were identified. So this is pretty impressive. And this, as a scientist myself, this gives me a lot of hope for the future. The fact that so rapidly after the discovery of this pandemic, after the discovery of this disease, we had sequenced it and made this information public to the world, really is part of what enables such a rapid development of vaccines for treating this illness. Subsequently, isolates were also sequenced with the Oxford Nanopore MinION sequencer, and the MinION sequencer has some very nice advantages for sequencing viruses like this. Primarily, as I explained earlier, the fact that the long reads pretty much remove the entire need for de novo assembly. So typically, when we de novo assemble a virus, we have to go through some process all-to-all mapping, filling out gaps, if there's any kind of low complexity regions, which isn't typically a problem with viruses because they have high information density. But if we have to do anything like that, then it's computationally and scientifically quite a challenge. Whereas long-read technology like Oxford Nanopore's MinION can just read an entire virus strand in one read in actually just a few minutes. So in the case of SARS-CoV-2 virus, which is 30,000 base pairs long, given that its ONT sequencer can read DNA at approximately 500 bases per second, it should only take 60 seconds for an entire, approximately, between one and say 3 minutes for an entire genome of SARS-CoV-2 to be sequenced and base called, which is extremely impressive. So nanopore sequencing is pretty unusual amongst the sequencing technologies. It doesn't work in the same way as PacBio or Illumina sequencing work. PacBio and Illumina sequencing essentially are forms of sequencing by synthesis. So the idea with PacBio and Illumina sequencing is that you synthesize new DNA through a polymerase reaction, and by using optical means, by essentially using fluorophore molecules, you can detect which DNA is being synthesized. Nanopore sequencing is totally different. Unlike PacBio and Illumina, which use optical technology, nanopore sequencing uses electrical technology, so semiconductor technology, to detect DNA. So how does this work? A protein nanopore is embedded inside a lipid biomembrane, and an electrical voltage is applied across this membrane. So when you have an electrical voltage across a biomembrane, and a biomembrane is a very good insulator, and you have a small nanopore inside it, a small hole, electrical current begins to pass through the nanopore. So the way that ONT sequencing works is that the DNA is passed through the nanopore via the assistance of a motor protein, and as the DNA passes through the nanopore, it actually interrupts the flow of electrical current. So if you can think about, in terms of in electrical terms, you can think about the protein nanopore as a resistor sitting on top of an insulator. So electrical current is passing through the nanopore, and as the DNA passes through, it begins to disrupt it. The underlying nucleotides are inferred by what's known as a DNA base caller. And a DNA base caller is nothing more than a piece of software which reads data from a sensor, let's say in this case, analog to digital converter. In the case of Illumina, it would be optical sensors, and converts that information into bases of DNA. The unique thing about the Oxford Nanopore base caller is that it depends entirely on deep learning and artificial intelligence. So it uses a seek-to-seek modeling, what's known as a seek-to-seek modeling process, which is very similar to those found in neural machine translation. So let's say you had an application which took human language, let's say me speaking right now, and translated it into written language. Let's say it was a Italian person speaking Italian. You recorded what they were saying at 3 kilohertz, and then you had a deep learning application which took that recorded Italian and translated into written Italian text. This is almost identical to how the Oxford Nanopore base caller works, except instead of translating spoken Italian recorded as an audio signal into written Italian, it takes the samples of electrical current passing through this nanopore and translates them into nucleotides, into a sequence of DNA nucleotides. And this entire process is totally deep learning based. Prior to deep learning approach, Oxford Nanopore actually used different techniques, biophysical modeling, hidden Markov model approaches. But what's remarkable is that the GPUs hugely outperformed these techniques, and deep learning improved accuracy very substantially by as much as 10 percentage points. And Oxford Nanopore sequencing nowadays is around 98% accurate, so really approaching the levels observed in other sequencing technologies like Illumina. Are there any questions about how nanopore sequencing works, and specifically around how base callers use deep learning to interpret DNA?
I don't see any question on the chat. So.
Okay, in that case, I'll crack on. So yeah, some advantages of this technology, and especially in the context of a pandemic, is that it's highly portable. So it's a portable and real-time device. It can be used in field. It doesn't have delicate optics, doesn't have delicate microscopy technology or anything like that, which is common to other optical-based sequencing technologies. It's entirely semiconductor-based. And in fact, it can even work upside down. It can work in handheld mode, and yeah, it is very portable. You can literally easily put it in your pocket. The MinION, which is Oxford Nanopore's probably most famous sequencer, is actually a USB device. So for anyone who's worked with NovaSeq or any sequencer like that, this is obviously quite a big differentiator in the sequencing technology. The fact that you've got a cheap device which can be easily transported and has the advantage of all these long reads, it is quite a remarkable difference from short read technology. That's not to say it's better than short read technology, just that it has certain advantages and of course disadvantages, like relatively lower accuracy rates to be considered. Portability is incredibly important. Probably the most famous example of portability being important in the case of DNA sequencing with Oxford Nanopore sequencers is that Oxford Nanopore have built the only DNA sequencer which can go to space. So the International Space Station, I believe currently does, and certainly in the past has done, had MinION sequencers on board. And these are used for experiments around identification of disease and diagnosis of disease. Let's say, for example, an astronaut gets sick. It's not easy to diagnose what particular bacteria, say, is affecting an astronaut in space. There's no hospitals there, there's no labs. And if you want to send a sample back to Earth, there's no easy way to do that. And so in this context, portability is extremely important. This is kind of an extreme example. There's not many places as remote as the and inaccessible as the International Space Station. However, if we're talking about pandemics, then obviously pandemics don't know any kind of boundaries. They don't know geographical, or, well, they know some geographical, but they certainly don't know any political boundaries. And so if you are, for example, sequencing the process of a pandemic, monitoring the process of a pandemic in more remote regions of the world, places like in the forested areas of West Africa or South America, then portability is very important. It's not easy to. So was there a question? Oh, I don't think. Okay. It's not easy to take a larger sequencer to these places, and yes, you can take samples and retrieve them and take them back to a lab in a more populated area, but this takes a lot of time. And in fact, this was particularly exemplified in the relatively recent Ebola epidemic in West Africa, where the MinION sequencer was really used very effectively to monitor the spread of the Ebola virus in West Africa by researchers there who would not have been able to do this with similar technology, and is similarly being used nowadays for the same purpose with the SARS-CoV-2 epidemic. Another big advantage of Oxford Nanopore sequencing for monitoring an RNA virus like SARS-CoV-2 is direct RNA sequencing. So when we sequence a virus, when we sequence an RNA virus, what we need to do is convert the RNA into cDNA, complementary DNA, for actual sequencing, because PacBio and Illumina sequencers have no ability to directly sequence RNA. And the issue with this is it introduces bias. So one nice feature of nanopore sequencing is that it allows a single molecule direct sequencing of RNA, and full-length transcripts can actually be characterized. Most excitingly is that, to my mind at least, is that epigenetic modifications can be understood. So we take, for example, this this trace over here. What we can see is two RNA reads, raw RNA reads, that is, both from SARS-CoV-2 in this case, both from the SARS-CoV-2 virus. So this is the same sequence of RNA. However, if we look at the actual current, so on the y-axis here is picoampere levels, so the amount of electricity, the amount of electrical current that's passing through a pore. What we can see is that for the same nucleotide, there's different electrical current levels, and what this represents is epigenetic modification. So things like methylation of particular bases. There's no way for non-nanopore sequencing to detect this, and it's quite possible that these kind of epigenetic modifications have a role in the infectiousness and the spread of the virus. So now I'm going to talk a little bit more about my specific area, which is the bioinformatics that we use for COVID-19 and how GPUs are used for this. So Oxford Nanopore, as previously mentioned, uses GPUs across their product line. So right now they have 3 commercially available sequencers, the MinION, the GridION, and PromethION. The MinION uses the TX2 GPU, and the GridION, the PromethION, use the V100 GPU. If anyone here is doing Oxford Nanopore sequencing and has issues with base calling, please do reach out to me afterwards. One nice feature of these GPUs is that they're not some special technology which is embedded into the instrument and can only be used in that very limited context. If you have raw data from these sequencers, then it's very much possible to bring them onto your machine and sequence them and do the base calling with your own GPUs. It's actually possible to use CPUs as well. However, this will normally be substantially slower. And why would we want to use GPUs at all anyway? Why do companies like Oxford Nanopore use GPUs for bioinformatics applications like DNA base calling? So GPUs are not that different from CPUs. At the end of the day, they're both microprocessors using caches and registers and the sort of same semiconductor ingredients that goes into making any microprocessor. But they are different in that GPUs are really designed for high throughput scientific and graphical computation. So CPUs are composed of a small number of cores, each of them having a very large cache and able to handle a small number of threads. And GPUs are composed, on the other hand, of thousands of cores, and they're able to process thousands and thousands of threads simultaneously. They are optimized for different things. So it's not that CPU designers don't know what they're doing and designing the wrong thing. CPUs are really optimized for latency. So if you're a computer programmer and you're writing an application to be deployed on, say, an operating system, let's say, I don't know, Microsoft Outlook or something like that, latency is really important. You want your application to be able to handle multiple different threads, and you want it to be very, very slick and very responsive. And this isn't what GPUs are for. GPUs are optimized for throughput. So something like DNA base calling, genome-wide association studies, stuff like that, where the throughput is really important, is really where GPUs shine. And GPU cores are typically simpler than CPU cores. So a CPU has a small number of very sophisticated cores, whereas a GPU has a very large number of much simpler cores. If we look at the DNA base calling example I gave earlier for Oxford Nanopore, going from the raw signals, electrical current to DNA bases is quite a non-trivial problem. And as I mentioned earlier, the state-of-the-art base callers all use deep learning now. So for Oxford Nanopore at least. So if you are interested in how base callers work and you want to see a real example of one, there is an open source project called Bonito, which is an implementation a very high accuracy of a very high accuracy base caller using the QuartzNet architecture, which was developed by NVIDIA for automatic speech recognition, actually, and it's totally open source. It's a DNA base calling is a topic I am very interested in, very much happy to talk about all day. So if anyone would like to discuss this with me later, then I'd be very happy to talk about it. But yeah, more generally, if this is something for your research or just for personal interest that you'd like to look into, do check out this link, github.com/nanoporetech/bonito, because it's a full implementation and there's everything you need to know about DNA base calling is included in that single repo. There's enough there for multiple PhDs worth of research. As mentioned earlier, the real advantage of that comes with portability of these Nanopore devices is understanding virus phylogeny. And so working with the RAPIDS team at NVIDIA, RAPIDS is a data science tool, we've been able to develop technology for understanding understanding ways that we can visualize and display virus phylogeny. And this is really important because if we want to understand things like where did a particular virus come from? You know, we all know it's SARS-CoV-2, but now we're hearing a lot about, for example, different variants. We're hearing about things like the UK variant and the, I believe, South African variant, stuff like that. The way we understand where these variants are coming from and the way we understand how they're spreading and the vectors they're using to spread is through construction of these phylogenetic trees, which is a very big active research area for NVIDIA. And this is also important because not only do we need to understand how the virus spreads, we also need to understand how well vaccines work on particular subtypes. When I gave this presentation first, this was kind of a very theoretical observation, but now we're actually starting to see concerns around, for example, the effectiveness of certain vaccines, like say the AstraZeneca vaccine, with particular variants, like the South African variant of SARS-CoV-2. So yeah, understanding the phylogeny and having very extensive sequencing programs the world over, not just in rich countries, is extremely important for helping us combat this and future pandemics. So I mentioned earlier that the initial virus was assembled using from short Illumina reads, and that this was a computationally challenging task. It's a very computationally challenging task, which is of particular interest to us at NVIDIA. So DNA is typically sequenced in very small fragments, and a full genome is assembled in the presence of sequencing errors, which is a very challenging problem. If there were no sequencing errors, then assembly would be still actually a challenging task, but a much reduced task because overlapping reads would be much simpler. So part of what we've done at NVIDIA to help solve this issue is we've taken a tool called RACON, and we've performed GPU acceleration of it. achieving acceleration of which is very substantial, going from 40 minutes on a V32 CPU instance down to 34 seconds for the best case scenario on a DGX2 instance. This is an open source tool called Racon, which we've accelerated using a partial order alignment algorithm, and it's available now. It's not especially useful for this very specific instance of understanding the this virus, because as I mentioned earlier, one of the big advantages of ONT sequencing for SARS-CoV-2 is that the long reads mean that the entire virus can be spread in can be read in one go. However, it is very scientifically interesting and very important in the more wide context of whole genome sequencing for humans. And the way this works is that first you start out with a bunch of DNA reads, you overlap the reads, and you can overlap the reads using GPUs using a tool we've developed called GoudaMapper. You construct a draft assembly, and then using the original reads and the draft assembly and the tool like Racon, which is GPU accelerated, you can produce the polished assembly. This is important because one thing that we understand, that we've learned about SARS-CoV-2 and COVID-19, is that it doesn't affect all people equally. So some patients with no underlying conditions experience very severe illness, and some patients who have who seem to be very vulnerable are totally asymptomatic, and we don't understand why this is. So what we need to do going forward is to start really pushing much more on association studies. We need to start building a much better understanding how a person's genotype relates to their phenotype, how susceptibility to a particular virus like SARS-CoV-2 is dictated by a person's genotype. And for this, whole genome sequencing and accurate whole genome sequencing, ideally de novo sequencing as default, is going to be absolutely crucial. The last thing I want to talk about is Nanopore, which is loop-mediated isothermal amplification for SARS-CoV-2 detection. And what this is a DNA amplification technology in combination with Oxford Nanopore sequencing, used for very accurate and cost-effective detection of SARS-CoV-2. So in the UK, this technology is actually now being used. So there's these vans driving around the country performing DNA, performing SARS-CoV-2 detection tests. And inside these vans, you have all the technology needed to perform LAMP amplification of samples, and DNA sequencers, ONT DNA sequencers, used to perform the detection to identify if SARS-CoV-2 is indeed present, and has been proven to be highly accurate for the detection in both symptomatic and asymptomatic population. So, as we know, for this particular virus, testing people is one of the main ways that we can stop its spread. People who have the virus do not need to self-isolate, and having a cheap and portable way to perform this kind of testing, and especially a type of testing which can give accurate and fast results. People get impatient if they have to wait, say, 10 days for their results, is really, really important. And so at NVIDIA, we're very proud that our technology in these sequencers, in addition to all the scientific advancements that have been made around things like, as mentioned earlier, deep learning for base calling, as well as alignment using GPUs and so on, is being used for this particular application. So that brings me to the end of my presentation. I hope you guys have found it interesting. I hope I've given you a flavor of the ways in which Oxford Nanopore sequencing, GPUs, and deep learning are being used in the world today to understand this virus, to help monitor its spread, and will be used in future with future pandemics. Because one thing I can say, I think one thing everyone can say with certainty is that this won't be the last time that we experience such a pandemic. So are there any questions? Hey, Vang, I can't hear you.
Yes. There's one question in the chat. How does the low accuracy on the ONT affect the final quality of the viral genome? We need to be able to reliably distinguish different variants of virus. For example, error rates lower than mutation rates.
Mm-hmm. Yeah. Okay, so that's a good question. So first of all, the accuracy is improving quite substantially. So as you can see from this chart over here, even between December 2019 and February 2020, there was about a 2% improvement in the accuracy, and this is all algorithmic improvements. So I think the accuracy now is actually pushing 99%. So the accuracy is not that low. It's pretty comparable nowadays to other sequencing technologies. But the person who asked this question does raise a good point. If the mutation rate is lower than, if the mutation rate is actually lower than the sequencing rate, well, what good is it? Because you can't, if you see a mutation, you have no reliable way of knowing which is it a true mutation or is it just a sequencing error? And the solution to that is this final step here, polished draft assembly. So the nice thing about Oxford Nanopore errors, and this is also the case mostly for PacBio as well, is that the errors tend to be very random. So what you do to resolve this error, these issues, is that you read the same DNA multiple times, and if you take it from the same sample, it should be the same DNA multiple times. And you perform what's called polishing using this technology like Racon, or there's other ones like Medaka, which I mentioned earlier. And by taking multiple reads and then averaging across them, and the averaging is rather complex, it uses partial order alignment, deep learning, stuff like that. But by averaging across them, you can reduce the error rate to well below the mutation rate. So it's now quite normal to get results of Q40, Q50, or even Q60. At the point when you get to Q60 with a virus that's 30,000 base pairs long, it's very reasonable to believe that any mutation you see is almost certainly real. If I don't know who asked this question because I can't see the chat, but if someone wants more detailed information about this question, I'd be very happy to take that by email and give you specific examples of the tools you can use to resolve this issue.
Yeah. I think the name on the chat is not clear, so I can't recognize or read who it is or who that person is. But yeah, thank you for the questions.
Okay.
Please go ahead. Any others would like to discuss? So the schedule for today is 2 hours, and the first hour will be Mike's presentation and discussion, and the next hour will be Emily's. But in case we can finish before that, then we can move up quickly and then finish the webinar sooner. But yeah, we have by schedule 15 minutes left, so please go ahead and discuss. And I don't know, the background of the attendees can be very diverse. So for someone from the biology background, this may be some familiar stuff, but someone from computer science, it could be something not so familiar. And then someone from computer science would like to see more in terms of how AI is used under the hood for this work. And I don't know, maybe that's that. I would like to make that a question. Then could you show a bit more something for the computer science side, Mike?
In what sense, Vang?
Is like for example, something about the construction of neural network or something like that, or algorithmic.
Yeah, I've got some slides of that. Just give me a couple of minutes. I'll just find some.
Yeah, sure.
In the meantime, if there's any questions on the chat, we'll be happy to take them.
Yeah. And usually I state upfront that this record is, I mean, this presentation is recorded, and everyone, if feeling comfortable, then they know that, and someone who doesn't want, then find a way to hide yourself. I mean, but yeah, just to notify that this presentation is recorded and shared later on a channel from Claudia. And I also would like to point out that this events and also series of events is co-organized by Claudia and AI for the People from Untitled University, and from NVIDIA as well. And at some point, we also have a third party co-organizers. Like in this case, we invite Emily from the University of Edinburgh.
Okay, Van, can you hear me?
Yes.
So you asked me to explain a little bit more about the computer science aspect of how a base caller works. And I've got this one slide over here, which is like one approach to writing a base caller. So let's say you had some DNA. Let's say you had some Oxford Nanopore raw reads. So under in this chart over here, the y-axis represents electrical current level, picoamperes, and the x-axis represents time in sample space. So just number of samples. And the actual dots represent labeled DNA. So how do we actually do this? The way we do this is we take how do we create this labeled data? We take an existing sequence, and we take an existing base caller, and then once we've base called it, we can normally achieve high enough accuracy to map it to a reference somewhere. And then once we've mapped it to a reference, we can get accurate data, because any errors which the original base caller made, we can mostly correct away. So now we have some accurately labeled data. And then what we do is you write a seek-to-seek model, which outputs conditional probabilities over time. Let's say this is called a conditional random field. And what the bottom chart shows is for the same data, the conditional random field, which a deep neural network outputs. And so what you can see, so these are run length encoded signals. So for example, one of them might be 4 A's, one of them might be 2 C's, and so on. But what we can see is that for most of the time, the actual deep learning algorithm is pretty confident in its predictions. It normally puts all the probability. So in this case here, the y-axis is a probability distribution, a multinomial or categorical probability distribution, and the x-axis is just the same as above, corresponding in sample space and time. So between what's interesting is that between 200 and 300 200 and 300 samples, what we can see is that the region where the probability distribution is confused. So the deep learning algorithm cannot decide what base is going on here. It's just very confused. And if we look at the sample, you can see that it actually is quite an ambiguous region. So the place where a human would have said this is hard to call, this is hard to base call, I would argue, is where the base caller itself is quite confident that it's a difficult region to base call. Because you've got this relatively flat sort of sloping region, it's alternating between two bases of DNA, and yeah, the deep learning algorithm gets confused. But essentially what you do is once you have this conditional random field, you write use Viterbi or beam search algorithm to create a path through it to decide what the underlying true sequence is. And this is fundamentally, I mentioned earlier, if you want to go on to, if you want to learn how a base caller works, there's some different approaches. This is one of the main ones. But this, if you were to write a base caller, this is fundamentally the way you'd approach it, which is actually a little bit different from the way in which traditional seek-to-seek, like automatic speech recognition systems work. They use something called they use something called CTC loss, which is a little bit different from this. You can also train a base caller using CTC loss, but this is an alternative and more intuitive approach, I would argue. So this slide pretty much tells you everything you need to know about how to write a base caller. But I do encourage anyone who's interested in this topic to go on GitHub and check out the Bonito project and check it out. And the author of that particular project, Chris Seymour, is a very responsive person. So if you have any questions, you'll get help, not just from him, but also from NVIDIA. I'll put my email address on. I'll put my email address on the chat. I'm always very willing to help with any kind of people to understand this technology.
Great, thank you, Mike. There's one more question here. Thanks a lot for the presentation. How are the error rates that the sequencing can be considered to be reliable?
In terms of raw error rates, anything from 90% upwards can normally be corrected quite easily. So I would say 90% is a reliable error rate. This is if we take, for example, PacBio's CCS data. Then this works by reading the same molecule of DNA multiple times. Each time is only around, well, I say only, it's still pretty good, but it's around 90% accurate. But once you exceed 90% accuracy, as long as the error rates are randomly distributed, which they are for PacBio and ONT, then this can be corrected quite straightforwardly. So I would say, to give a number to your question, I would say 90%.
I mean, 90% accuracy.
Single read, yeah. And then after that, you will need to do polishing of multiple reads of the same region to correct those errors.
Thank you.
Okay.
Yeah. Anyone, please go ahead and post the questions. This is very interesting and also precious opportunities, I would say, to talk with someone very experienced from NVIDIA. So please go ahead, and if you have any question regarding running the sequencers and also the GPUs or analyze the data, this is the time and opportunity for you to throw up the questions.
If not, I'll be very, like I was saying, if anyone has any questions they want to mull over, I'm very responsive over email. But otherwise, maybe we can take a 5-minute break before Emily's.
I think I would say, yeah, 3 minutes. It's 5 minutes to be exactly start in Denmark time is 2 p.m. But yeah, let's say 5 minutes later, we will start. So yeah, see you.
Okay, see you in 5 minutes.
Yeah.
All right, let's start for the next presentation. Hi, Emily, are you there?
Yeah, I'm here.
Hello, Emily.
Can you hear me? Hello?
Yeah, we can hear you, Emily.
Okay, great.
I don't hear Emily yet.
I can hear Emily.
Yes, I think in the meantime, I would like to introduce Emily. So just a second. Let's share the screen. Hi, Emily. Just to confirm, are you there?
Yeah, I'm here.
Then maybe check the sound if it is working.
I can hear Emily just fine.
Oh, then it's something with my with my speaker. Then just a second.
Speaker, yes, I think it should work now. Sorry, I was using the headphone, and when I walked out, it's disconnected. Sorry for that. Yep. All right. So the second presentation today is PangoLEARN practical classifiers on the scale of the pandemic. And probably if Emily decide to change the title a bit to fit better, that's also fine. But some information about Emily. So she is the person behind the machine learning implementation of the PangoLEARN tool, and that's called PangoLEARN. It's an improved version of PangoLEARN, which the first one didn't have the machine learning parts. And Emily is currently the postdoc at the Institute of Evolutionary Biology, University of Edinburgh, and she holds a PhD in computational biology from the School of Informatics, the University of Edinburgh. And before that, she works as the software engineer at Amazon, and she got a bachelor degree in computer science at Johns Hopkins University in the state. And I'm very excited to have you here today, Emily, because I have been also using PangoLEARN and so build a workflow around PangoLEARN and PangoLEARN for Denmark COVID team. So please go ahead. The screen is yours.
Great, thanks. Thanks for that introduction. I will just share my screen.
Yeah, I will stop sharing then. Let me see.
Okay. Okay.
Yeah. Yeah,
right.
you should be able to share the screen now.
Let me see, sorry. It's always right. Okay. Right. It doesn't seem to want to let me share my screen. Oh no, now it is.
Yeah.
Okay, can you see my screen now? Okay, great.
Yes, I do.
So I will, great, so I can get started. Right, so thanks, Ving, for that introduction, and thanks, Mike, for that presentation about nanopore sequencing. We're certainly using nanopore sequencing at the University of Edinburgh, and also I work as part of the Arctic network, and we also have sort of protocols for sequencing, and we do use Oxford nanopore sequences for that, quite a lot of minions, and then at the University of Edinburgh we have a quite big GridION also. Right, so my presentation is called PangoLEARN, Practical Classifiers on the Scale of the Pandemic. I'm going to talk a little bit about something that Mike went into just a little bit, which is phylogenetics. And I'm going to give a kind of introduction of what phylogenetics is and how we use it, and specifically how we use it in the current pandemic. And then I'll talk a little bit about PangoLEARN and PangoLEARN, which Vang just mentioned. So I mean, one of the big questions that maybe you're left with after Mike's presentation is, okay, so we have this amazing technology that is quite portable, that we can send all over the world, that we can use to sequence COVID genomes. And this is a screenshot of the GISAID homepage from last night, showing that there had been, so GISAID is one of these big public repositories for coronavirus sequences and also for flu sequences. And as of last night, there had been 601 submissions to GISAID. So 601,000 coronavirus sequences that have been submitted since the beginning of the pandemic. But that's all great, but what do you actually do with all of those sequences? And there are a bunch of answers to that question. So for example, Mike mentioned vaccine production, and there's also drug discovery, but I'm going to focus on this other thing that he mentioned, which is tracking coronavirus variants and using sequences that are being taken throughout a community to understand the underlying sequences of transmission that are happening within that community. So this is a simplified version of a phylogenetic tree. So when we talk about phylogenetics, this is something that basically all revolves around this idea of the phylogenetic tree. So basically, it's a representation of a whole population of virus sequences, or it could be anything, it could be any organism, and then how they're related together. So I'll quickly explain how this works. So this axis, the y-axis, doesn't actually have any meaning. There's no particular reason why virus 1 isn't where virus 2 is and virus 2 isn't where virus 1 is, or this whole bit of the tree could be over here. The axis that matters is this x-axis, and that could represent either genetic change. So if up here, the root node represented the original coronavirus sequence in Wuhan, the further away from that sequence a particular virus is, that would mean that it would have more mutations compared to the root sequence. Or this axis could represent time, but actually those two concepts, so they sound very different, are kind of the same in phylogenetics, because as it turns out, there's this concept of a molecular clock. So each kind of virus species has a relatively steady rate of mutation. For the coronavirus, for the current coronavirus pandemic, that's about 2 to 3 mutations per month, which is actually quite slow. for a virus, it's about 30% slower than, for example, Ebola. But that basically gives you a very easy way to convert genetic change to time. So this axis could be either. And each of these leaf nodes or tips is what we normally call them, represents an individual patient. So this is virus 1 would be a specific sequence that was taken from a patient and you know that we've then sequenced. So and so would virus 2 and virus 3 and virus 4, etc. But the internal nodes here represent our kind of best guess at the most recent common ancestor of some subset of these viruses. So this node here would represent our best guess at when there was an ancestor virus that then led to virus 1 and virus 2. So that's all great. You can imagine that, you know, if you were to catch every single case of coronavirus and sequence it, then you could have this really comprehensive tree that would give you a lot of information. So you would know exactly when the first person got sick, exactly who they gave it to and when, and you could very easily kind of manage your pandemic. But unfortunately, that's not really how it works. And it's especially not how it works in a virus like the current pandemic, because of the number of people that just don't know that they're sick, that don't know that they've been exposed. So it's really difficult to kind of construct that. And this slide is just to kind of show why that's so difficult. So you can say the red dots are transmission events. So someone kind of taking their virus and giving it to someone else. Each of the diamonds are a mutation, and each of these blue dots with a letter indicate a sampled case. And just from this, so if this was the ideal tree where you would capture every single case, this is maybe what it would look like. But for example, if there's no mutation between two sampled cases, we couldn't necessarily link the transmission at all. There's no way for us to say, okay, well, you know, a particular virus is exactly the same as another. So there's no kind of story that we can tell there. So that's a big problem. And that's, you know, while it's quite good that the coronavirus, they kind of mutate a little bit slowly, that's good for things like vaccine efficacy and immune response. It's not super useful for building these phylogenetic trees. And actually, it's a bit easier to do that for something like Ebola that has a higher mutation rate. Additionally, there are mutations that we might miss because we never sample them. And then there's also, I mean, there can be kind of whole chains of transmission that we're missing. So we're really basically trying to take a very imperfect snapshot of all of the virus that exists in a community and then try and rebuild this as perfect as possible tree that represents this transmission train. And then we can get these kind of very useful, we can ask these very useful questions like who started or what process kind of started a particular outbreak in a school or a hospital, or what kind of restrictions would be helpful? Should we be restricting travel? Should we be restricting schools? What kind of decisions do we need to make locally and at kind of a government level? So how do we build these trees? Well, then building a tree is very important, and building, you know, making this really good guess of reconstructing that process is really important. So one potential method of doing this, which I'm sure you'll all realize is the terrible idea, is just to build all of the possible trees and compare them all, find the one that's most likely. Unfortunately, these two exclamation points aren't there because I'm just really excited. It's because it's a double factorial. So the number of trees grows really, really quickly, where n is the number of sequences. So as of last night, if we have 601,000 sequences, you can imagine this is just a completely ridiculously large number. And even for a much smaller epidemic, like, you know, in Ebola, I keep going back to Ebola, but that kind of thing, it was still already just way too big of a number. So one of the easiest algorithms that people use is the neighbor joining algorithm, where basically you find the two most similar sequences, you make them into a little subtree, and then you treat that tree as another sequence, and then you keep doing that. So then you join that to another tree and so on and so on. That's a really common algorithm to use, and that's because it's quite fast. But building just one tree gives you kind of a really poor probability of finding a really ideal tree, something that's a really good guess. So that's not something that's kind of good enough for our current use. So this picture here is from Holyrood Park in Edinburgh. It's basically a big collapsed volcano that's in the middle of the city. And I have it here just because I wanted to quickly mention that the main way that we kind of build these trees is using a hill climbing approach, which I'm sure will be really familiar to a lot of the people in the audience that have kind of a computer science background. But basically you start at the bottom of the hill, and you can imagine that the whole entire hill is kind of this whole tree space, this whole area that you have to look for the ideal tree. And you take a step from your particular location, you take the step that is leads to the highest spot that you have only one step away from you. And you keep doing this until you reach kind of a maximum, until all of the steps that you can take would lead you somewhere down the hill or down the level of probability. And that's a pretty good approach. I have this picture here because you can kind of imagine that, well, actually Holyrood Park has multiple maximum points, and there's many local maxima. So you can kind of imagine that you could end up here, you could end up here, or occasionally you might end up at the top tree. And that's a limitation of this approach. And actually this is quite a big area of research. I'm not going to go into all of the different tree building algorithms, but there are kind of ways that people try and kind of maximize their likelihood of ending up at the kind of true maximum. But this is kind of a limitation of this approach, and that means that actually these giant phylogenetic trees that we're building are kind of brittle. Every time that you build a tree, you might end up with a slightly different tree, actually. And that means that all of the kind of work that we're doing, we have to kind of know that the underlying structure that we have is somewhat unstable. So let's say that you found the perfect tree building algorithm, and then you have this really massive tree. This is actually an old picture of a tree that was only, I think, about 300,000 tips. So there's quite a lot there that you can't even see. Now we're building much, much bigger trees. So how do you actually turn this into useful information? How do you take this tree that you've built and actually sort of monitor variants of concern, or discover variants of concern, or track a local outbreak in a hospital? So one really useful thing that you need to do is you need to kind of break this tree up into usable labels. So there are a bunch of different approaches, and you might have seen them. There's been quite a lot of news about coronavirus nomenclature recently, a few like Guardian articles and things like that about what we actually name these variants and what that means. So the next strain approach, and Mike actually showed a screenshot of next strain also. So it's quite a commonly used workbench and set of tools. They have these kind of really high level clades. So that's these two columns, or these clades, where they have these kind of really large groupings of viruses. And this, I should say, this is quite an out of date image. This is from a couple of months ago. So there's now actually several more clades, but they're quite large groups. And they're quite useful, but mostly for things like tracking trends in a whole community over a long period of time. And then we have kind of this other approach, which is the PangoLEARN approach, or the Pango lineage approach, which is to have a hierarchical representation of the tree split into what we call lineages. And I'll go into what a lineage actually is in a second. But our approach, the key to it is that it's hierarchical and it's quite fine-grained. So we can look at very, very specific, very local outbreaks. And the reason that we want to do this is that's really what you need to do to kind of do real-time outbreak investigation. And if you want to know a little bit more about the nomenclature and these naming systems, we have a publication here that was published in the middle of last year that goes a little bit more into the thought process. And actually, there's quite a lot more being done right now to kind of rewrite these rules that I'll get into in a minute. Right. So what is a lineage? How do we kind of decide on these categories of sequences? So a lineage is a group basically of somehow related sequences that are separated from the rest of the community context by some kind of epidemiological event. So you can imagine that maybe someone comes from another country. So maybe someone comes from Brazil and goes to Denmark, and then kind of maybe spreads around their sequence, you know, to a couple of people that they come in contact with. Well, that's something that we would see very easily on a phylogenetic tree. We would see something come up that wasn't at all like the other sequences that we were seeing in the community. And that would be represented as a relatively long line separating that group of sequences from the rest. And then we would see maybe a cluster appear, a bunch of sequences that were kind of disseminated and that came from that original kind of epidemiological event. So that's what we categorize as a lineage. And how we actually find these lineages is something that I'm going to go into in a second. So as I said, all of these lineages are hierarchical. So we have the original A lineage, which is from Wuhan, and we have, you know, the original sequence would have been the first person that was infected. And then we have, there was some kind of event that led to the B lineages. And that has some history in China before it then came to Europe, and actually the B.1 lineage, which seeded most of the epidemic in Europe, came around the same time as that original outbreak in Italy. So there was some kind of transfer event. There was some kind of mutation so that we could actually identify that there was a different sequence that was emerging. And then we could see that sequence actually take over. So the vast majority of sequences that we get now are children of the B.1 lineages. So you maybe have heard of B.117, which is the UK variant, the South African variant, and also the Brazilian variant. All of these kind of variants that you're hearing about that have many different names, those are all kind of coming from this B.1 variant quite a while ago. And so this might get a bit confusing. So one of the rules that we have is we have a maximum depth of naming. So once you get past a certain number of dots, we then give it an alias. So that's why, for example, the Brazilian variant, which we've named P.1, is actually a descendant of B.1. It's just that it got pretty far down, and so we at that point say, okay, this has a new name, just to make things a little bit easier to pronounce. Right. So you have your big phylogenetic tree. You have all of these labels. What does that actually mean to you as a researcher? So if you're sitting in your hospital or your school and you've had an outbreak and you've sequenced all of these patients, how do you actually learn from the system that we've put into place? So that's the question that we're trying to answer with our tool PangoLEARN. So PangoLEARN, which is a acronym for Phylogenetic Assignment of Named Global Outbreak Lineages, is a tool where basically you input your sequences and will give you an assignment. So it's a classifier. We'll tell you what category you're in. And then you can look at this wider kind of phylogenetic tree and say, okay, well, this particular lineage is associated with these clinical characteristics, it's associated with this travel history, and then you can use that information to kind of make some decisions. So for example, if you're worried about the occurrence of the B.117, the UK variant occurring in Denmark, you could use this tool to tell you how many of the sequences in your population are of this variant. So the original approach, which was written kind of right as I joined the group, so I wasn't on this project yet, was a maximum likelihood approach. So basically we would take this really massive tree and we would make a guide tree. So we would really downsample this tree so that we would have this kind of skeleton structure. And then they would compare any of the kind of query sequences against that tree, so that then they would give you an output of, okay, this is the most likely class. And this was done by Anya O'Toole and JT McCrone, who are still really helpful on this project. And Anya O'Toole is still the one that's doing quite a lot of the kind of building of the Pango lineage rules at the moment. And I'll talk a bit about that in a minute. The problem with this approach, I mean, it was very accurate. I think it had about a 98% accuracy and a similar recall. So it was a very good performance. But even at the beginning of the pandemic, so this was done around April and May, the creation of the guide tree took a very, very long time, both in terms of like human hours and in terms of computational time. And then also the actual assignment took a really long time as well. It was taking, despite the tree being way, way smaller than it is now, something like, 10,000, 20,000 sequences, it was still taking on the order of hours to assign a group of sequences. So that's when PangoLEARN became PangoLEARN, and we decided to take a machine learning approach, and that's when I was kind of started working on the project. So the requirements for this kind of model that we were building were quite stringent. So we needed to have a very, very fast training time. This model is trained sometimes every day, sometimes multiple times a day. So it can't take a week. It can't be a deep learning approach that's going to really take up all of our servers for a very long time, because we're already doing that, trying to build all of these phylogenetic trees every day and that kind of thing. So it really needed to be kind of lightweight and very fast. It needed to be very accurate. We didn't want to lose any of the accuracy of the maximum likelihood approach. So something around 98% accuracy. And we needed something, the assignment time had to be very fast. So one thing around this time, one of our collaborators, Anthony Underwood, and his team built a web application for PangoLEARN. And we were already kind of having significant queue times to submit sequences. So we really needed this assignment time to also be really quite quick. And it really needed to be scalable. And this time, those exclamation points are just exclamation points, because when we started working on this, our training set was something about 25,000 sequences, and now it is over 600,000 sequences. And we still need to train the same models, and we still need to get the same quality of results. So seeing all of these requirements, I think, was quite intimidating to me about a year ago when I started working on this. So, but the good news and the spoiler alert is that we did it. So, so the training set. So as I'm sure all of you machine learning people know, 90% of the problem is having a great training set. So that's something that we've had to work with a very large number of people to do. And we have, as you know, both me and Mike have talked about, we have a very large network of people around the world that are doing sequencing, and that amount of sequencing that's happening is only increasing. So something that's quite important is that sequencing is not, the amount of sequencing is not uniform across countries, which then kind of limits this approach. But that's something that we can talk about a bit later. Right, so we have this very large amount of sequencing, and we're still trying to build a very up-to-date real-time tree. So that's something that Ben Jackson is working on. He builds a new phylogenetic tree using some kind of custom approaches pretty much every day now. And then that tree is still, it's semi-automatically and semi-manually curated. So we have a basically someone that occasionally goes through and looks at the tree and tries to identify whether or not some new identified lineages are legitimately new lineages. This is something that's currently being done mostly by AnioTool and also a bit by Verity Hill, but it's something that is becoming kind of a more international collaboration because a lot of the sequencing is being done locally by people that are very interested in our lineage assignment system and really want to be able to say, okay, I have a bunch of sequences coming in Wisconsin that look like a new lineage. And so it's kind of become a crowdsourced thing that, and then also it's become, we're trying now to separate this kind of process of building a training set from the process of then assigning a classification. So we're kind of separating the assignation and the designation, meaning that we're trying to make these kind of tree rules that are kind of completely, you know, they're kind of, there are rules that are, trying to think of, I've completely lost the right word. So there are rules that are not quite set in stone that are being offered by the model that are kind of suggested tools, suggested rules. And then there are rules that are, meaning there are lineages in the tree that have been kind of set in stone since the beginning of the pandemic. So at the moment, you know, when we build a new tree, we're not changing anything in the A lineages or even in the kind of early B lineages. Really where the changes are happening are at the kind of tips of the tree. So we're trying to kind of separate those rules so that we have a set of lineages that are set in stone, and then we have kind of ones that might change every time we build a tree. And that's become the kind of Pango lineages, the ones that are quite set in stone. And then the PangoLEARN and PangoLEARN kind of system is the bit that just kind of offers rules at the offers lineages at the end of the tree. I don't know that that was the most clear explanation, but I'm happy to answer more questions about that. It's kind of an ongoing process and something that we haven't completely sorted out yet. So, okay, so we have this training set. Basically, we have 600,000 plus sequences that are all being assigned a lineage through this kind of semi-manual, semi-automatic curation system. So we have very luckily, we have a very good quality training set. And this is the part where I pretend as if I had a plan going into this. When I first got on the project, it was really something where we weren't expecting it to be quite such a difficult thing to do. We were thinking, okay, well, this maximum likelihood approach is a bit slow. So let's see if we can kind of throw some simple model at it and see if it helps. So the first version of PangoLEARN used a very simple multi-class logistic regression. Something that's quite notable about that is there is actually no, as I'm sure all the machine learning people know, but for people who might not know, there's no concept of hierarchy in a logistic regression, despite the fact that we know that the data itself is quite hierarchical because we've made it be hierarchical. We were using a model that really had no concept of that, but actually it worked really quite well. I have some statistics on the next slide, but basically it had a 96.5% accuracy, I think, in that first round. And the main problem with the logistic regression was actually training time. So at the very beginning, when we had 30,000 sequences, it would train in a few hours and everything was fine and dandy. After a couple of months, suddenly it couldn't actually run. It was just too much data and it completely would die as soon as I tried to run it. And that's when I tried using a decision tree approach, a very, very simple idea. Just basically, you can think of a decision tree as kind of a flow chart where you have rules where you start at the root and you kind of move down the tree, making decisions at every point until you reach at the end a node that represents a classification. I made this decision because I thought, well, okay, the logistic regression can't keep up with the data. Let's try actually harnessing the hierarchy and see if that can kind of improve things. And it did actually. So the decision tree, even as it stands with 600 plus sequences, it only takes about 2 hours to train. The problems with the decision tree are that it's quite brittle. So we already have a tree structure underneath that we're trying to represent that's quite unstable. And so then on top of it, a decision tree is itself quite unstable. So you can change one kind of small bit of the tree and you could end up with a completely different decision tree. And then that might, for example, if you had a group of sequences that were all being classified in a particular group, suddenly we update the model and they're all being put in a slightly different group, and that's something that we've had problems with, and that's actually why but it's one of the reasons why we're trying to kind of separate the kind of potential rules, prospective rules, from those that we know are really set in stone when it comes to this decision tree. So we have a set of lineages that aren't going to change, are not unstable, are not brittle, and then we have some that are a bit more unstable, and you kind of have to be aware that if you get one of these classifications, it might slightly change in the kind of short term before it finally settles on its final lineage. And a lot of these problems are solved using a random forest. So I was really expecting kind of the random forest model to be the best kind of model to use. So I built a variety of different random forests. So a random forest is basically a collection of decision trees, and it kind of democratizes the process. So you kind of get rid of a lot of that brittleness, a lot of that instability, because you might have changes in 1 or 2 of the trees, but you're not necessarily going to have changes in all 100 of the trees when you build a random forest. I will now show you some statistics that will show actually we didn't really gain that much using a random forest, and we lost quite a lot in training time. So This first panel here shows the training time. So this is the logistic regression, and you can see it started with a pretty reasonable training time, and then you get here and it stopped running and it wouldn't run anymore. And that was, you know, kind of in the middle of last year. And so you can imagine it would definitely not want to run just now. So then we have a random forest with 1,000 constituents, and I've just kind of simplified the random forest to kind of the two that gave us the best results. So the 1,000 constituent random forest was actually increasing quite a lot in training time, and I wasn't. This chart kind of ends late last year, and I'm not positive now that it would even run just now. The decision tree and the 100 constituent random forest had a kind of an equivalent training time. Really not very much. I mean, including all the pre-processing again, like a couple of hours. So you have to look at the scale here. So the recall rate was pretty great across the board, to be honest. And the F1 score was also pretty great across the board. We were having pretty good model fit, which makes sense because, you know, a decision tree, a random forest, they really do represent hierarchical data quite well. And looking at this kind of data, really what the decision was, well, we're losing a bit in terms of training time with the random forest. We aren't really gaining much in terms of performance. Let's just stick with the decision tree, especially because we know that we're not going to be able to get rid of all of that instability anyway, because ultimately it's just unstable tree data. That's what we're dealing with here. So that's actually what the current version uses, is just a decision tree, which I know sounds shockingly simplistic. But I mean, I built quite a lot of models, and that's what we came up with. So there's some limitations to this, though. As I said, there's quite a lot of instability in decision trees and already instability in the underlying data. This is something that we're kind of managing more using the kind of lineage rule system that we're building than actually the model, because the model really does a very good job of representing the underlying data, and most of the instability comes, it seems to be coming from actually the kind of phylogenetic tree building part of it instead of this end of things. And then one of the big limitations that we are trying to correct is limited compatibility with ambiguous and missing data. So something that Mike mentioned, and there were a few questions about, I saw, was about kind of the error rate in Nanopore sequencing. I mean, the error rate, as he said, is quite, you know, is pretty low and is getting better, but there is always some level of errors in sequencing data. And when you get errors in sequencing data, you can get things like ambiguity. So you can get a site where basically the base caller doesn't know what's there. You also get missing data. So for example, we might have some areas of the world that are much better sampled than others and that kind of thing, and that can kind of bias the model as well. So what the future direction is, and what I'm currently working on when I'm not on other projects and hopping back and forth, is I want to basically harness the power of the decision tree. So clearly that's a really great representation, a really efficient representation of the data, but it's not very flexible and it's brittle, as I've said. So what I want to do is basically build a hierarchical logistic regression, something analogous to kind of to hierarchical softmax and NLP, that would basically make all of these decision points kind of probabilistic questions that you can then kind of better incorporate ambiguous data into, because then you can have some kind of probabilistic poll instead of a categorical you know, question that you can't answer based on missing data. Right, so just to kind of give you some places where you can look for a little bit more information. So we have this Cove Lineages website, covelineages.org, that will you can go to if you have questions about kind of what all of these lineages are, how we come up with them, and, you know, kind of the clinical implications of them. So if you want more information about kind of B117 or the South African variant or any of these kinds of things, we have a lot of that information there. There's the PangoLEARN web app, as I mentioned, that was built by Anthony Underwood and his team at pangolin.cog/-uk.io. And that is a really great, useful tool. And you can basically upload your sequences and it'll give you an answer quite quickly of lineage assignments. And then we have the command line tool. So we get quite a lot of people submitting very large number of sequences, like 5,000 sequences in one go on the web tool. If you're going to do that many sequences at once, I would recommend you use the command line tool. It'll probably get back to you faster, to be honest. And that's available on GitHub. So this is just the rest of the Rambo group. So they've all been at some point or other working on this project. So thank you to all of them. And yeah, that's me. I'm ready for any questions if anyone has any.
Thank you, Emily. That's interesting. So everybody, please go ahead and ask questions. I actually have a couple of them, so I will give priority to others first. So there's someone, yeah, Mike actually asked, are your team using GPUs for the non-deep learning approach, like decision trees?
Yeah, so sometimes and sometimes not. So the real answer is we actually haven't really needed them. So we have, we were using GPUs when we were doing the logistic regression for sure, and we really did need them. For the decision tree, I mean, running in like a single thread on a CPU, it works just fine. And that's something like my background in my PhD was like epigenetics and kind of using language models to kind of represent epigenetics and kind of really putting some GPUs to work. So I was really surprised that we didn't actually need it. And it's surprising that something that has been so simple has really been the answer, because I was really expecting to have to kind of use some kind of deep learning solution using lots of hardware. But actually, it really hasn't been necessary. Certainly in the kind of future directions, I would expect we will need to use them. And we do have access to quite a few of them that we use all the time for building the phylogenetic trees. So our group is definitely using a lot of GPUs, but not for the decision tree.
Mm-hmm. Thank you. Someone raised the hand. Ray Sieber. Please go ahead. I'm sorry for the name pronunciation.
Oh yeah, it's Raphael Sieber.
Yeah.
I'm from Denmark, from Statens Serum Institute.
Yeah.
And I just wanted to ask you, you referred again and again to that the underlying data actually is unstable, and that's why the predictions are also unstable. The data we have sequenced in the past is basically, that's what it is, no? So what happened years ago is not going to change.
Yes. So that's something I maybe didn't make this point very clear. So we're trying to so kind of, yes, old sequences, something more than even say a month or maybe even a couple of weeks in some circumstances. That's not unstable. That's something that, the kind of lineage designations that we've had a chance to look at for a while, we're not expecting that to change. Really, when you're kind of rebuilding when you're rebuilding a tree, the changes are quite incremental. So there's something, like lots of polytomies and homoplasies that can kind of mess up where things are placed kind of towards the tips in the tree. So if you certainly if you submit something that's in a B.1 lineage or in an A lineage or something like that, that's definitely not unstable. But something maybe from a brand new lineage that we've kind of discovered yesterday, that's going to kind of wiggle around a bit. And that's kind of something that's exactly why we're trying to kind of separate this like assignation step and the designation step. So kind of the creation of this training set, there are parts of it that are going to never change again, because we've stared, like we're not expecting to find new original Wuhan sequences anymore. We're expecting, there have been a few different variants that have kind of taken hold. Like, for example, in Europe, the B.1 lineages completely took hold, and that is still really like the majority of sequences that we're seeing. So it is quite rare to see any A lineage, and certainly not an A or an A.1. So that kind of part of the tree is really set in stone. And that's kind of been set in stone by, smart virologists that know what they're talking about and have decided that is what the tree should look like. And it's really the things kind of at the end of the tree. So, the B.1.1.1, many steps down the tree that are unstable.
Okay, thank you very much.
Thank you. The next question is from Joan Poton Gomez. Please.
Yeah, okay. Good evening, everyone. I am working in Colombia, and right now we're facing in maybe 2 months, we will have the third wave, okay? The Brazilian lineage have come to Bogota. I want to know if we can use that tool to prevent that lineage multiply here in Bogota.
Yeah, so I mean, the tool itself, it'll just tell you what's available in the community. But certainly, if you are doing community sequencing, so if you have, when people are getting tested, when they're going to hospital, if they're having the viral genome sequenced, the tool will tell you what proportion of cases in the country, like if you're keeping track of the assignments from all of these cases, you can easily figure out what proportion of cases in the country are the Brazilian lineage, the P.1. And on top of that, you can kind of, and what people are using it for is to, when they get people tested, they're also doing track and trace. They're asking kind of these epidemiological questions of all of the patients, like, where did you come from? Or where do you go to school? Or who have you been in contact with? All these kinds of things. So then you can maybe kind of come up with policies to say, okay, well, this is the better restriction to have. Like, for example, after the UK's first lockdown last year, we had very little coronavirus circulating in the country. So we knew that there was very little virus. And then once the country opened up again and there was quite a lot of travel, it was very easy for us to see, actually, all of the virus circulating right now is a Spanish lineage. And it's coming from people going on holiday and coming back and then sending their kid to school without self-isolating and all these kinds of things. And there was a report that we published, I think at the end of last year, that I can send you to that has a bit more information about that. But basically, yeah, like by combining this kind of tool, phylogenetic trees, and specifically our PangoLEARN tool, you would be able to take all of your local sequences, upload it to the tool. We could tell you what lineage it is, and then we could tell you kind of where that's coming from. And then you can combine that with these kind of questions to come up with sensible policies that would help you kind of hopefully control that third wave. Yeah, it's a little bit complicated because, of course, we don't know absolutely everything about these lineages. So P.1, we know, for example, there's a little bit of immune escape, so that there's evidence that there's a little bit of an increase of serial infections, more so than with other variants of the virus. So there's things like this that we're still kind of learning, but certainly it's incredibly useful to actually know what kind of sequences you have in your community. And that's kind of a big, a lot of countries have no idea what's circulating in their community, and that makes things a lot harder for us to kind of actually track, what's actually going on with the virus's evolution.
And we don't have enough resources because it's too expensive. It seems that we are checking only 1% of the entire population, and you are 7%. Is that okay?
Yeah,
right?
I mean, yeah. So at the moment, I think we're actually increasing that number at the moment. But yeah, right now we're super sequencing approximately 1 in 10 cases. Denmark, I think, is doing even better. I don't remember off the top of my head. Some countries like New Zealand and Australia are sequencing 100%. Certainly, the more that you do, the easier it is. If you so one of the things that the Arctic Network does is it kind of helps other groups set up sequencing. So if you want, you can maybe email me and I can maybe get you in touch with some people that are. Yeah, I'll post my email to the chat. So I'll just do that right now. So it might be that we can kind of set up some kind of collaboration, and we do have a fair number of collaborations in I think like 7 or 8 countries at least right now that I know of. So that would be something that would be really interesting to set up. And it's kind of good for everyone because it's really good to know. For example, there's very little sequencing happening in my country, in the US, and so we get quite a lot of sequences where maybe there's a bit of travel history and maybe they're coming from the US and they have kind of quite a long, line, quite a lot of mutations compared to what's happening in our community, and we kind of have to kind of piece that information together after the fact. Yeah.
And the last thing, can you share this video because I want to that other scientists can see it again, watch it again.
Yeah, sure. I will send out an email in a couple of days to share and if also the slides with everyone, and so you get the information. Yeah, thank you, George. And
Yes.
Any other questions, please? I have a quick question then. For someone who wants to play with like improving machine learning or try other algorithms, I haven't got time to check it, but how about getting the training set and then?
Yeah, so we published the training set.
Yeah.
It's on one of our GitHub repos. I think it's code support. I can find a link to that afterward, and I can send that to you. But yeah, the training set is available on GitHub. So you're welcome to give it a go. Yeah, I mean the bar I guess is somewhat high in that we do need really kind of high 90s in terms of accuracy, and we do need it to train quite quickly because it's something that we're kind of automatically running every day. But yeah, you're welcome to have at it.
Yeah. So for the tree construction, the decision tree constructions, then which the under the hood, which algorithm was used to construct the tree? Because I think that there's a different among several tree constructions algorithm.
Yeah, so it kind of changes periodically. At the moment, I think so that's code that was written by Ben Jackson in the group, and I think at the moment he's using IQ tree. They're kind of something that we're working on is obviously most of these tree building algorithms, they weren't built for something quite this big. So they take a really long time, and sometimes, I mean, you run it for ages and it doesn't give you what you need and that kind of thing. So something that we're working on a lot in the group is kind of how to take a tree that we think is quite reliable and kind of build onto it incrementally. So that's something that Ben and Rachel, 2 other postdocs in the group, are working on. So hopefully, I mean, all of that will be public source. So hopefully that will be available. There's also a tool called Scher that already does something similar. So you might want to look into that.
Yeah. Okay. Thank you, Mike. He's dropped off now. But yeah. Okay. Well, thank you, Emily. It was a very interesting talk, and I think we have a couple of minutes left. So if any quick questions, please go ahead, or I will have some wrap up for today.
How much time do you take to develop that solution?
So there's a lot of different pieces to it. So there's the kind of the wider pipeline. So there's a lot of kind of data processing that needs to happen before you actually get to the kind of classification step. And that's been in development and kind of continues to be improved, but that's been in development since kind of the beginning of the pandemic and around April. But the kind of core of it was actually built quite quickly. So I think the whole thing was kind of up and running starting in May, approximately, and then certainly the kind of first PangoLEARN versions were available like May and June. So it's been around for actually quite a while on the scale of, you know, the kind of year of COVID. It's been around for a while.
Yes. Right. We have a couple of minutes left, so I would like to wrap up for today. And I would like to say that this event we do every month with different topics in the, we say the series name is Interesting Topics in AI. And then if you have any thing that you want to discuss or have it on the next topic, then in the survey or the feedback form that I send out later, please put it in the feedback form. Then we will try to make it happen. So thank you, Emily, and also Mike already left, but thank you for the two speakers today, and also thank you everyone for attending, and look forward to see you next month. Thanks.